Hello, I'm going to explain today's lab how to measure the absorbance of your standard solution. Okay, so the standard solution is a phosphate solution and it's called I just put A, B, C, D, E, F and you have it in a test tube. Everybody has A, B, C, D, E, F, G. So there are 1, 2, 3, 4, 5, 6, 7 tubes. So we are going to measure 7 samples first, okay? So the nature of their standard solution. Nature of the chemical is a phosphate ion. Okay, phosphate ions are in this solution A, B, C, D, E, F, G. Why are they A, B, C, D, E, F, G? It's because they have a different concentration of phosphate and it's written in your procedure. So it's from 0, 0.5, 1, 2, 4, 6, 8 micromolar, yeah, of phosphate solution is inside this solution. Okay, so we are going to use spectrophotometer to measure these solutions. We have done the spectrophotometer before. We are measuring the absorbance. We call this optical density or absorbance, yeah, um, of each solution. So A, B, C, D, E, F, G. Okay, so we're gonna write 0 to, yeah, 0 0.5, 1, 2, 4, 6, 8 micromolar. Okay, and then you will get a value, yeah, like this. It is possible you might get a data like this because the concentration of phosphate is increasing, so you will get a higher absorbance in as you go up the concentration. So you might get a concentration like this if we plot the graph of the absorbance. Yeah, so you might get a graph like this. Okay. So let's call this a standard graph, yeah, or standard curve. Okay. So first, your task is to measure the absorbance of the solution A to G. Okay. So how do you do it? First, you set the nanometer. We are checking UV absorbance. So check the nanometer, set it to 650 nanometer. Okay. If the machine doesn't go to the exact 650, just set it close to 650. Yeah, so it depends on the machine. Sometimes the machine doesn't increase, by 10, or if you can set whatever you can set close to 650. Yeah, maybe some machine cannot go to 650, you can set it at 645, yeah, whichever. So depending on your machine's capability, just set it close to the 650. Okay, that's our objective. After that, we use a cuvette. We have to put our solution inside the cuvette, right? So you need to prepare your cuvette, take the cuvette out of the box, yeah. So the first thing is we need to set the zero, right? The 0 point, yeah. So 0 is without any phosphate. Yeah, so to measure this, we actually use a detection agent. Detection agent. So the method is you have to add the detection agent plus the thing you want to measure. Yeah, so in this case, ABCD. Yeah, right. So first we have to, let these two react first, then after that we measure. Okay, so I have written in your procedure that use 1 ml of detection agent and put 2 ml of the phosphate or the standard solution. I can name this as a sample, generally sample. So the sample should be 2 ml and the detection agent will be 1 ml, So let it react for a little while. So wait for few minutes, Um, the best will be 4 minutes. If you could wait 4 minutes before you measure the absorbance, that way we can get the best result. So take note of that. Okay, so you have to measure, A, B, C, D, E, F, G. Okay, so prepare cubed, A, B, C, D, E, F, G, that many cubed, and put 1 ml, 1 ml of um, detection agent. I will provide you with the detection agent, and then you put 2 ml of either A or B, or C or D. Uh, then wait for, if you could wait, let it react. Then we measure, then we can find out how much phosphate is inside the solution. So we will get a data like this. Okay. Okay. Now the next part two of the experiment is we have two beakers. We are going to fill the beaker with some water, distilled water. The method is written on your procedure, so please refer to the procedure. So it will tell you how much water to put, and we are going to put a living organism in here. We have freshwater mussels. We have pondweed. Okay. So what we created is an artificial, water system. So it can be a pond or lake, or it can be a river. We just made an artificial water system. And then what we're gonna do is we are going to add some pollution to it. We are going to add pollution. Our pollution is a soapy water. Soapy water, because soapy water contains phosphate. So just now we learned how to detect phosphate, right? So we are going to find out if mussels or pondweed can reduce this phosphate. So since we have limited time, what we can do is after we add the soapy water, the pollution, right? We will take out certain amount, and put it in the cubed, and then we call this 0 minute. And then 10 minutes later, 20 minutes later, we're gonna take one more out. 10 minutes later, we'll take out one more. 20 minutes, we take out one more. 30 minutes later, we take out one more. 40 minutes later, we take out one more. Okay? And measure the phosphate ion using spectrophotometer. And then, I don't know, maybe in the beginning, there may be this amount of phosphate, and maybe muscle may be taken up. They can take it into their cells. Pondweed can also take it into their cells. So we don't know, maybe phosphate level will stay the same or maybe they will start to go down. We don't know what's gonna happen, yeah, but we will find that out, yeah. So let's try this experiment, yeah, and then we can discuss, yeah, how to interpret this data and how to find out the exact concentration of the phosphate, yeah, in this artificial system. Okay, so I hope this makes sense. And next lab, we will have the actual data. Then we will use Excel spreadsheet, yeah, and then we will learn how to plot the graph, and we will learn how to plot this standard curve, yeah, and we can also calculate the slope of this curve, and then you will get a y equal ax plus minus b equation. Yeah, once we have this equation, right, uh, if you get a data from here, you got certain absorbance, right? So let's say, oh, we got the absorbance of 0.01, yeah, but we don't know how much phosphate is inside here. So we go to this graph, and then the x, we do not know the x, yeah, we do not know the x-axis, but we have the y value, which is the absorbance. So you plug it in here and we solve, yeah, this is the algebra. We will use algebraic equation and solve for the x, yeah, and then um, you can actually find out how much phosphate is in your sample. Okay, so I will teach you how to do this using, yeah, Excel or Google Sheet. Yeah, any of those graphing app can be used. Yeah, so let's do that next time. All right, thank you.